아이스크림 사랑 두근두근 설사 달고면서 레일 살살 녹아들어 행복한 순간 빠져드는 맛 다가와 느껴봐.
The objective of vaccination. Our bodies have two types of immune system, which are innate immune system and adaptive immune system. When we get infection, the innate immune system is activated first, and then the innate immune system further activates the adaptive immune system to get rid of the pathogens. The adaptive immune system is specific and it also has memory. So, the objective of vaccination is to activate adaptive immune system in order to get the prophylactic immunity in our bodies before getting infection. So, when we got infection, we already have the preformed immunity to fight with pathogen. Therefore, we won't get.
When microorganisms enter to our bodies, they have three main steps to generate infection or disease. First is attachment, second is colonization, and third is invasion or releasing of microbial toxin. The production of high-efficiency vaccine is to prevent at the first step, which is the attachment. If the microbes can attack to the host cell membrane, they cannot produce disease. But if the vaccine that prevents at attachment step can protect, next step should be prevent at colonization step, and if this step doesn't work again, the next must be the immune response that can prevent microbial invasion or toxin neutralization. We call the immunity which we receive before getting an infection is prophylactic immunity by using weak microbes or part of that pathogen to activate immune response.
The story of vaccination started first in China about 500 AD. The Chinese used pus from the lesion of smallpox patients and inoculated to healthy person, and this method was called malleolation. There are also some evidence show that blowing off the powder caps into nasal cavity could raise the immune response against smallpox. About 200 years later, in 1717, a British lady named Lady Mary Wortley Montagu. She learned about malleolation from Constantinople, Turkey, and when she came back, she introduced the method by using small amounts of smallpox pus scratched into the healthy person's arms. In 1796, there was a hero named Edward Jenner. He found that a mermaid who was infected by cowpox would not get smallpox, so he set an experiment by using pus from a mermaid named Sarah Nims and inoculated to the 5th person's arm. He got a mild symptom. When he got well, Edward Jenner inoculated smallpox pus to 5th, and nothing harmful happened. The method developed by Edward Jenner spread over, but sadly, some people didn't believe in this. Then in 1836, Dr. John C. Martin tried to find a new method by using fluid from people who died from smallpox, inoculated to cows either, and used the pus from lesion injected to people, and found that the result was the same as Edward Jenner's. In 1948, he introduced the vaccine using live vaccinia virus, which is in the same family as smallpox virus, but less virulence. Finally, in 1980, smallpox was eradicated according to the WHO. The discovery of other vaccines in 1870, Louis Pasteur worked on the attenuation of the chicken cholera. Then, in 1879, he produced and developed vaccine for chicken cholera by injecting living bacteria to the chicken, and they all died. After that, he told his assistant to inoculate fresh prepared bacteria to the chickens before holiday, but he forgot. A month later, he returned and injected it, but that bacteria was too weak, so the chicken had only a mild symptom and they became healthy again. After that, this group of chickens were again injected with active, fresh bacteria. Luckily, the chicken didn't have any sign of symptoms and they didn't die. This vaccine was discovered by chance, thanks to his assistant for the mistake. In 1885, rabies vaccine was used in humans and first administered by Louis Pasteur. Pasteur successfully prevented rabies in 9-year-old Joseph Meister. Meister's mother brought the boy severely bitten by a rabid dog to Pasteur in hopes of preventing it. Pasteur felt that the boy would die from rabies infection if he did nothing, so he decided to help by giving that boy the course of 13 injections, one each day, of vaccine made from rabies virus infected nerve tissue of rabbit. Meister never developed rabies, and the incident was regarded as a success. Well, there have been many types of vaccine developed afterwards. You may want to know how many types of vaccine now. Let's watch this to make you more understand. Development of vaccine. From the discovery of vaccination by Edward Jenner, many kinds of vaccine are developed. The first type of vaccine discovered was live attenuated vaccine, which is made of living microbes by using the less vigorous strain or make the pathogenic strain less potent by inoculate those microbes to an animal which isn't its natural host, or using embryonic aid. I repeat, surpasses several times until that microbes decrease their violence and can't cause pathogenesis. Example of live attenuated vaccine is vaccinia vaccine, OPV, and BCG for tuberculosis. The advantages of this type of vaccine: first, they activate the immune response both HIR and CMIR. Second, no need to use adjuvants, and third, no booster is required. And the disadvantages of this type of vaccine are unstable, less heat tolerance, and can be reversed to violent microbes. Plus, there are some disadvantages of live attenuated vaccine, so the new development of vaccine is come to the consideration, and that is killed vaccine or inactivated vaccine. The kill microbe is used. Many methods have been used to kill the organisms, for example, heat, chemicals like phenol, acetone, alcohol, formalin. Kill microbe still have immunogenicity but no pathogenicity. Example of kill vaccine are rabies vaccine, influenza vaccine, and typhoid vaccine. The advantages of this vaccine are more stable and safer than live vaccine. Bacteria or virus in the vaccine can cause disease, so the vaccine can be given to immunocompromised persons. The disadvantages of this vaccine are that it requires adjuvant and need to boost many times. Since live attenuated vaccine and inactivated or killed vaccines sometimes are not the most satisfying vaccine for us, therefore the scientists are still looking for the new one which is safer, more stable, and provides higher efficacy like subunit vaccines. The vaccines which use only some part of the pathogen or organism to activate the immune system. The advantages of the subunit vaccines are: the first, can be given to immunocompromised person; second, appears to give long-lived immunity; and third, the adverse reactions are very low because only parts of the pathogen are used. The disadvantages are must be given several doses to generate lifelong immunity, and appropriate adjuvants are needed. Subunit vaccine can be separated into three types. First, toxoid vaccine. Second, recombinant vaccines, and third, DNA vaccine. Toxoid vaccines are vaccines that make from toxins or harmful secreted biochemicals from bacteria. This vaccine is used when bacterial toxin is the main cause of it. Toxoid, in general, is an inactivated form of toxin. There are many ways to inactivate toxins such as 0.5% sodium hypochlorite or formaldehyde that used in tetanus toxoid preparation. Examples of toxoid vaccine are tetanus vaccine and diphtheria vaccine. The advantage of this vaccine is only inactivated toxin is used, not the whole organisms, so the disease cannot be occurred, and the disadvantages is this technique is limited to the disease that caused by toxin-producing bacteria. Recombinant vaccine are the genetic engineered peptide vaccines. Processes in synthesizing are as follows. Interesting gene region is selected and inserted into the vector of choice, and then given to the bacteria or yeast to synthesize the peptide, which is called recombinant protein, and this is used as vaccine. Examples are hepatitis B vaccine and HPV or human papillomavirus vaccine. The advantage of this technique is that it can produce a large amount of vaccine, and the disadvantage is that it requires a high sophisticated technology, so the cost of production is high. DNA vaccines are the vaccines that using DNA encoded for the specific protein antigen which can activate the immune response of the host to be used as vaccine. The interesting region of DNA is selected and amplified by PCR, then inserted into the chosen viral capsid like the vaccinia virus. Many kinds of DNA from different pathogens can be inserted together in one capsid. So, then the immune response to many kinds of pathogens can be achieved. Next, the viral capsid with selected DNA nucleotide is injected into the host by intramuscular injection. When the DNA vaccine is injected into the host cell, the capsid will be removed, and DNA will enter into the host cell. The host cell then produces viral protein, so APC can uptake the peptides encoded from the particular DNA, then an immune response is activated.
Process of vaccine production. Do you know how a vaccine can be produced? And do you know how many checkpoints that the vaccine has to pass before it is used with humans? You do not have to keep you curious anymore, because today we will tell you the standard process of vaccine production. To make you understand easily, please imagine as I say. The steps of vaccine production are only two. The first is laboratory and animal studies, which is focused on finding the new vaccine and try to test it with small animals, for example mouse or big animal like monkey, but not human. And the second is clinical studies in human subjects. As they name, this step focuses on testing the efficiency and adverse reaction in humans. That's the concept of it. For the laboratory and animal studies, this can be divided into two stages, which are exploratory stage and preclinical stage. The exploratory stage is the stage that involves basic laboratory research for antigen, which will be used in the new vaccine. Scientists identify the natural or synthetic antigens, find some parts of bacteria or virus, which can be used to prevent or treat disease. These stages mostly take about 2 to 4 years. If the scientists had already found the antigens, the researcher would continue to the next stage. That is the preclinical stage. This stage is testing the safety of candidate vaccine, the immunogenicity, and the ability to stimulate our immune response by using cell cultures, animals such as mice, monkey, and gibbon, which its immune response is close to human. They may also suggest a safe starting dose for the next phase of research, as well as a safe method of administering the vaccine. These stages also done the challenge studies, which is infecting vaccinated animals by the target pathogens. After the first step investigational new drug, you approve the candidate vaccine and give the permission to do the next step. The second step, clinical study with human subjects, has 4 phases, and there are phase 1, 2, 3 vaccine trials and phase 4 trials. Phase 1 vaccine trials. This phase use small groups of adults, about 20 to 80 subjects, and if the vaccine is pursued, researchers will test with adults first, then gradually step down to a target age. In this stage, subjects know whether vaccine or placebo is used, also known as open label. The goal of this phase is to assess the safety of candidate vaccines and find out the type of vaccine. For next, phase 2 trials, this phase use more subjects than the first phase, and that is about 100 subjects, and in this group of subjects, it may include the subjects who are at risk of the disease. This test is a randomized with placebo included. The goal of these trials is to study the candidate vaccine's safety, immunogenicity, dose, and route to delivery. The successful of candidate vaccine will move to the next step, phase 3 vaccine trials. In this test, 100 to 1,000 subjects are included in this study. At this stage, subjects will not know what they have got, whether it's a vaccine or placebo, so we call it double blindness. The goal of this trial is to study the vaccine safety in the large group of people and hold that a rare side effect which cannot found in small groups may be found. After complete the third phase, the investigator will submit for the biological license application to the Food and Drugs Administration or FDA. FDA will inspect the factory where the vaccine will be made. After licensure, FDA continue to monitor the facility and also control for the purity and efficacy vaccine. and flow to the last phase, phase 4 trials. This phase of testing vaccines after it is released to the market by the drug company.
EPI vaccine or expanded program of immunization vaccine. The goal of the EPI is to have every eligible child fully immunized with efficacious vaccines. Nowadays, Thailand has 8 vaccines that is compulsory by Ministry of Public Health on EPI to give every children. There are BCG, HB, DTP with HB, OPV, MMR, JE, DTP, and DT. And in Timor-Leste, there are BCG, OPV0, tetravalent, DPT or Hep B, measles, and TT.